Welcome to part 4 on chapter 11, looking at nucleic acid structure, DNA replication, and chromosome structure. So far we have looked at the identification of the genetic material in part 1. We looked at molecular structure of DNA and RNA in part 2. Part 3 we focused on DNA replication. Part 4 we're going to look and see how this DNA is actually condensed down into chromosomes so that we can fit it into our cells. So we're going to look at eukaryotic chromosomes, and this is because prokaryotic chromosomes, they're just a circular piece of DNA. Eukaryotic chromosomes, they are linear, so they have a beginning and an end to them, and they're usually discrete from each other, separate from each other. So a typical eukaryotic chromosome, if you stretch out the DNA, it would be 1 meter long. And we have to fit that chromosome into a cell that's only 10 to 100 micrometers across. And not only that, eukaryotic chromosomes in our cells, we have 46 chromosomes. So all 46 of those have to fit into these teeny tiny little cells. So we're going to look and see how that actually can be possible. So a chromosome, it's a discrete unit of genetic material, and we'll see some images of chromosomes in this chapter and the next chapter. So chromosomes are composed of something called chromatin. And this chromatin, it's made up of proteins and DNA. If you remember from the first part of this chapter, we had that experiment where the scientists wanted to determine if proteins or the DNA held that genetic information. So this is where we're going to put those proteins and DNA back together into folding out these chromosomes. So there's 3 levels of DNA compaction or folding. So the first one is called DNA wrapping. So the DNA is wrapped around proteins called histones. And there's 8 histones that come together. So 8 of these proteins come together. We have a DNA molecule that wraps around those 8 histones, and that structure together forms what's called a nucleosome. So if you look at the image here, the dark pink is our DNA strand, our DNA double helix strand. The light pink are those histone proteins. So we have 8 histone proteins, they come together, and then we have our DNA wraps around almost 2 times around one of these histone protein complexes. And that DNA is anywhere from 146 to 147 nucleotides. So about 150 nucleotides get wrapped around 8 of these histone proteins. There's green linker proteins that help to hold that DNA onto the histones. And then this complex is called a nucleosome. Okay. So you get all this DNA wrapped around to form these nucleosomes. Those nucleosomes, they then zigzag back and forth to form a 30 nanometer fiber. And it's called a 30 nanometer fiber because it's 30 nanometers wide. And the current model suggests that the nucleosomes, they zigzag back and forth from each other. And you can see that in the image right here. That 30 nanometer fiber that's shown here, it then is going to form radial loop domains. So now the pink molecule is a 30 nanometer fiber. So this 30 nanometer fiber, it interacts with the nuclear matrix, which is proteins inside of the nucleus that help to organize all of the DNA and chromosomes. So we get these radial loop domains that form, and they're attached to proteins inside of that nucleus that make up the nuclear matrix. When the cells prepare to divide, these chromosomes will compact down even more. Euchromatin is when it's not as compact. So here we have an image of a nucleus inside of a cell. The euchromatin, you can kind of see white spaces. The DNA is just forming loops. Heterochromatin is when the DNA really compacts down right before the cell wants to divide. And that's when we get the chromosomes that we think of when we think of chromosomes. So they're going to be discrete units that we can actually see within the nucleus. So to kind of review, we have our DNA double helix up at the top. That DNA gets wrapped around histone proteins to form nucleosomes. And if you look at the image from a microscope on the left-hand side, it looks like beads on a string. So each of those beads is a nucleosome. And then the DNA is the string part of it. Those nucleosomes, they zigzag back and forth to form our 30 nanometer fiber, which is 30 nanometers wide. That 30 nanometer fiber interacts with the nuclear matrix to form our radial loop domains. Those radial loop domains will form bigger loops, so we'll end up with heterochromatin that'll condense down into the chromosome at the bottom. And that's the chromosome that we generally think of when we think of chromosomes. So these chromosomes are chock full of DNA wrapped around all these proteins, folded all together in these loops, and really, really condensed. Okay, so this chapter looked at kind of the identification of our DNA. We looked at molecular structure of DNA and the RNA. Part 3, we looked at DNA replication, and then very briefly we looked at chromosome structure in this section.